All right. Good morning, everybody. My name is Herbert Schiller. I'm affiliated with the Helmholtz Zentrum Munich, and it's my pleasure to introduce today Emanuel Wyler, who will give today a lecture about his investigations on COVID-19 pathogenesis in hamster animal models. Emanuel did his studies, his Master of Biochemistry at the University of Zurich, followed by a PhD in biochemistry at ETH Zurich and a postdoc at ETH Zurich, and now he's currently a senior postdoc at the MDC Berlin in the Landthaler lab. As you can see in this slide here, the Lifetime Conference 2.0 is already open for registration, so we wanted to make you aware of this. And it's a pleasure to introduce myself. So I'm giving the lecture in 2 weeks on June 4th, 9:30. And with this, the usual information, so you can raise your hand for questions. You can ask your questions during the talk in the chat. The lecture will be recorded, and I think with this we are looking forward to your talk, Emanuel.
Good, thank you very much. I hope you can see it. Yeah. Thank you very much, Herbert, for this kind of introduction. And I would like also, I'm very grateful and honored that the Omics Network has me here. I would also like to thank Marco Uhrig, Mara Kika, and Nina Gasparoni for their huge organizational work in the background. So today I will talk about that topic that keeps us all busy at the moment in various aspects, COVID-19. We look at COVID-19 pathogenesis in hamster animal models. This is, as you can see here, collaboration of many different people, mainly from these three institutions, from the Free University in Berlin, the Charité University Hospital, so from respiratory medicine and infectious diseases. There are like two parts, a long part which covers the single cell analysis of the Syrian hamster, which is out as a preprint on here. And in the second part, I will talk about the Roborowski Dwarf Hamster, which is not yet published, but we are hoping to write this up soon. So a brief introduction about what I will talk about in COVID-19. I mean, the topic is well known to most of you. So the basic issue here is that there is in this very nice plot, you can see this is at -5. You see the time of infection, and time 0 is kind of the symptom onset. And at this time, people start to have perhaps a bit of fever or coughing or so, but it's very unusual that people feel heavily sick at this moment. And the severe and critical illness that affects some 5 to 10% of people only starts something like 10 to 14 days after infection, or 1 week after symptom onsets. This is then when you have this transition from, most people have this mild to moderate illness, so the symptoms disappear, people get healthy again after about 2 weeks. But some people take this detrimental route to severe illness and critical illness. This means shortness of breath, which is usually the first symptom of a more severe disease course, which also requires hospitalization and eventually also ventilation, and also invasive ventilation. The reason you can see here on this right picture, so these are scanning images of a human lung of a COVID-19 patient. So this is a front view, and where it's dark, it's kind of healthy tissue, but this so-called ground glass behavior here, or appearance here, is whitish sections, or in the cross section here. And here, this means that in these parts, the lung starts to fill up with liquid, with immune cells and so on, and this leads to the shortness of breath and makes ventilation necessary. And we are interested in our work, essentially, what happens here? What is it that makes like these 5 to 20% of people severely sick, so that take this route here, compared to most people which experience a mild to moderate illness? So we are kind of on this section here interested. Why do we need an animal model for COVID-19? So COVID-19 deteriorates rapidly, so over the course of days. in patients. However, when we have, and this is what I've explained before, if we have patients in hospital, it's usually several days after infection. So at the time point where likely the kind of the trigger of the critical disease course has already started. It's easy to sample also cells for single cell sequencing from blood. Lung sampling is usually only done for severe cases. So these bronchoalveolar lavages. So you put in a little bit of salty liquid into the lung and flush out a lot of cells, and they can be also analyzed using single cell sequencing. This has been extensively done. But this is also only possible for severe cases, because moderate cases, they usually don't have invasive ventilation, and then these lavages are kind of ethically not possible. So what is not accessible from patients, we cannot get moderate disease in the lung, as explained before. We do not get a full picture of the lung because these lavages only get soluble cells. So like macrophages, monocytes, B cells, T cells, but to only a very limited amount cells, which are our tissue resident, so epithelial or endothelial cells. We also don't see a full temporal development because usually the first week or the first 10 of days of infection, people are not in the hospital and we cannot see what's going on then there. And also it's very difficult to get samples from outside the respiratory tract. For example, investigations on heart biopsies, also single cell driven, but usually it's not possible to look at samples from outside the respiratory tract in patients. This can be done in autopsies, but this is a different story. So this is where the animal model comes in, because we can cover the entire course of disease, really from the first days or hours of infection until the development of severe disease. This has been now mostly done with hamsters, and the reason is that hamsters, in contrast to mice, can be infected with the standard SARS-CoV-2. And so here are first of the initial papers from last year. This is now more or less exactly a year ago on Syrian hamsters. So there are different kinds of hamster. Syrian hamsters are the golden hamsters, like the middle hamster, because they have like mid-size, but later last year, also dwarf hamster came into focus, and especially this Roborowski Dwarf Hamster, which I will cover in the last part of this talk, which is a highly susceptible model for a rapid and fatal course of SARS-CoV-2 infection. So this one here, the golden hamster, has a moderate disease course, and the Roborowski Dwarf Hamster has a severe to critical course of disease. And this means we have two relatively closely related animals which have a completely different course of disease. And so our idea was, if we now compare in detail these two animal species, perhaps we get some indications why in 80% of people SARS-CoV-2 infection leads to moderate disease and in the others to severe and critical disease. On a side note, since we are in the single cell omics network, I would like to emphasize this. I think you have seen this many times, that single cell omics was really key in COVID-19 research. I think it was yet another breakthrough year for single cell technology. Of course, single cell and single nucleus sequencing was heavily used in bronchoalveolar lavages from lung and blood, what I mentioned before, but also heart in myocarditis patients and so on. And what is also been extensively done is single nucleus sequencing from autopsy samples from all tissues. Then CyTOF has been employed several times, which is more kind of a protein-driven approach, mostly on blood samples, on peripheral blood mononuclear cells. CyTOF is also constantly being used to look at surface markers, especially in the context of immunity, and then also immune repertoire profiling, such as B and T cell receptor sequences. So omics helps us here to follow the disease over time and disease severity. So it's also this key thought of the Lifetime Initiative, for example, brought to action here. Good, the content today, I will talk about the disease course in Syrian hamster, the longitudinal cell type composition, all our analysis that we have done in there. And then in the last part, I will cover the initial analysis of the data from the Roborowski Dwarf Hamster. So this Photobus Roborowski, and this other one is this golden hamster or Syrian hamster. Good, how does the COVID-19 infection in Syrian hamsters looks like? So at day 0, hamsters are infected with 100,000 PFU, so infectious units of SARS-CoV-2. And over the course of about 7 days here, they lose something like 7 to 10% of their weight. And this is an indication of of whether the hamster is sick. So there's no temperature measurement, so no recording of coughing or so. It's the weight that is key here. But as you can see here, they recover then quickly afterwards. And at day 10 or so, they're more or less healthy again. This goes along with a very quick clearance of the virus. So this is how much virus is there per gram of lung. And I would like to emphasize it's a log scale. There is a lot of virus here at day 2 and day 3 post-infection. But at day 5, you see, for example, these 3 hamsters here, they have about 1,000 times less than before, and here it was not detectable anymore. Here with viral copies, it's a bit better to see. But you can see there is this dramatic drop already at day 5, and then at day 14, there is also only some remnants of RNA that can still be detected. This is how the lung looks like. This is naive hamsters, and I would like to emphasize day 5 here. You see a lot of this is this hematocycline and eosin staining. You see a lot of these dark patches here, which is an indication of immigration of immune cells and so on. And this is kind of also by the pathologist analyzed as some sort of inflammation of the lung. And as you can see here, this peaks at day 5 when the virus has already started to clear out. This also supports, and this we have also seen that COVID-19 is to a large degree also an overreaction of the immune system. So here, this is to quantify this, so the pathologists have some scoring systems, and then they can assign some score to the lung, and you can see the lung inflammation score is highest at day 5. Compared to here, where there's kind of a lot of virus, but not so much inflammation. And here there is a lot of inflammation, which actually also impairs the lung function, but not so much virus anymore. Good. What was the data collection that we did on these Syrian hamsters? Of course, we got naive hamsters, so uninfected, and then the first time points were day 2, day 3, and day 5, and then day 14 in the end. We assessed the virology and the histopathology. Of course, this is what I have already shown. We did bulk proteomics of both lung and blood, and bulk RNA sequencing of lung and blood, and then of course also the single cell RNA sequencing. I will mostly cover the data from the lung here. Good. How does the cell type composition in the lung look like? This is just to confirm that we get all known cell types of the lung out there. So we had these lung pieces which were enzymatically digested for 15 minutes at 37 degrees to kind of reduce, as you know, this stress response that comes up after infection, after this enzymatic digestion. And we have like all kinds of lung cells here, a lot of monocytic macrophages, alveolar macrophages, and interstitial macrophages, monocyte, neutrophils, and B cells and T cells. And here on the left side are the tissue cells from the endothelium, smooth muscles, fibroblasts, and epithelial cells. So 81 and 82 cells. The epithelial cells are the one lining the bronchial and alveolar compartments of the lung. And 82 cells are also thought to be the prime target of productive replication in the lung, because they express this famous ACE2 protein, which is the entry site for the virus. This here on the right side is the cell composition in percent. As you can see here, this is always day 0, day 2, day 3, day 5, day 14. You have an increase of macrophages, alveolar macrophages at day 2, and then monocytic macrophages, they come up a bit later, but this is temporal, and then it goes down again. And on the other side, you can see, and this I will come back to that later, for example, you have a strong increase in T cells and natural killer cells on day 5. Questions in COVID-19 pathogenesis. So what is known? I mean, lot is known now. I mean, within this 1.5 years, we have seen tremendous progress in understanding this relatively complex disease, but some key aspects are still unknown. So in most people, the infection is likely local in the airways and self-containing, and in moderate cases, there is lung damage, but self-resolving. This is what we see in the Syrian hamster. And this is in severe critical cases, there is a non-contained inflammatory response that on the ICUs requires invasive ventilation. And this is what we can see in these Roborowski Dwarf hamsters. Effects on the tissues outside the respiratory tract, I don't cover this, but I just want to mention it briefly, are generally thought to be more like a consequence of cytokine signaling. So this famous cytokine storm I would like to emphasize that this is under heavy dispute. So some people say there is a lot of virus also outside the respiratory tract, and other people say there's absolutely nothing. I think an exception is the olfactory bulb, which is, I think, now settled that there is some virus migrating through the nose into the olfactory bulb and maybe in some proximal parts of the brain. So what do we want to research now? So the first part of looking at COVID-19 pathogenesis is, where does the disease start, in which cells, at which time points, and by which trigger? And of course, if we're talking of trigger, what is directly triggered by some virus infection, what is secondary? Because in a virus infection, like for all pathogens, we have a lot of cytokine signaling and so on going on. And the second part is what amplifies disease, which factors contribute to amplification and limitation. So some things that are under discussion here are, for example, autoantibodies against interferons. So this is a very busy slide, but I only want to show, kind of look at the big picture here. So these are gene expression changes per cell type, per time point. So we have 2, 3, 5, and 14 days. Without looking at the genes in detail, you can clearly see that you have this very strong response at day 5. So this is where most gene expression are actually happening. And at day 2 and day 3, you only have like some initial things. So this is where most of the transcriptional changes happen relatively late. And I would like to remind you, day 5 is the day when we have the virus already being, to a large degree, being removed, but where we are at the peak of the inflammation. The strongest transcriptional response is not by epithelial cells, which are the primary targets inside of replication, but by macrophages. So here on the left side, I skipped this because time is, I think, running out a bit. But on the left side, I would like to explain this in detail. So we see here at different time points, day 0, 2, 3, 5, and 14, we see how many AT2 cells are expressing this ISG15, which is a standard interferon-stimulated gene and also triggered upon direct infection of the cells. And on this side here, we have the monocytic macrophages. And as you can see here, kind of all of the monocytic macrophages start to express this gene, in contrast to the AT2 cells. So the cells in which are actually directly infected by the virus, they have a relatively moderate response. And this is particularly true for CXCL10, which is an important pro-inflammatory cytokine. And as you can see here, macrophages start to express this at day 2 and day 3, and then also have very high expression of this pro-inflammatory gene at day 5. So we think that this means that the actual drivers of this hyperinflammatory response are clearly not the epithelial cells, which are the primary target of infection, but the macrophages, which sense the virus, but in which there is likely no productive replication. So this is how the pathology looks like. I go over this briefly. So here we have a bronchus, and everything in red is viral RNA. So we have a lot of infected bronchial cells here. This on B, this is now alveoli, like these compartments here. We have 2 different types of cells, the AT1, which are more the flat ones here, and the AT2, which are more the kind of rectangle-like cells here. And as you can see, most of these epithelial cells are actually not infected. Here on the other side, we have macrophages. We have macrophages that have engulfed other cells, but have also macrophages. So for example, this is a macrophage which has taken up a lot of virus, but not a cell. So macrophages take up the virus. We know from in vitro experiments that the virus doesn't replicate in macrophages, but it's being taken up. And as you can see here, a lot of macrophages here. So this is an alveolus filled with macrophages. So this invasion of the immune cells is how this looks like under the microscope. And many of them have taken up some viral particles. If they take up the viral particles, what do they do? So I would go here only on the right part of the slide. So this one here. And we now can distinguish, of course, between cells which contain the virus, which is, for example, this one here. This one here. And this bar here indicates these are macrophages or day 2 that do not contain the virus. And as you can clearly see, at all time points, and particularly already at day 2, if a macrophage takes up a virus, a lot of, for example, CXCL10 expression is being triggered. So more cells start to express CXCL10 and also their expression values are much higher. So take home message here is 82 cells, they don't do much when they take up a virus and when the virus productively replicates. This is also because in these 82 cells, the virus starts to express all its factors that suppress interferon signaling. And we can now look at in a more systematic way. So which are the pathways, which are the genes that are induced upon take-up of the virus. And this means if there is a big red dot, means that these genes here, and these are all pro-inflammatory cytokines, they are triggered when the virus takes up, when the macrophage takes up a virus. And all these genes here and some others are downstream of TLR and NF-kappa B infection macrophages. So what you think here is that there is a clear and very strong activation of the TLR signaling leading to NF-kappa B activation, and this leads to the strong pro-inflammatory response that is triggered by macrophages. This is per se not a problem if it can be contained, but that is the issue in severe and critical COVID-19. You have this strong pro-inflammatory response that is not contained. And we think that this data shows that it clearly starts at these macrophages. They take up the virus, they get a very strong TLR and NF-kappa B activation, and they trigger then this strong pro-inflammatory response. We also looked at the endothelium and cytotoxic cells, and this is perhaps interesting from terms of single cell data analysis here. So what have we looked at here? So as you know, there is not always, if you have a single cell, it's not always a single cell. You all know this, could be doublets. And what we have seen is that particularly at day 5, we have kind of cells, so kind of cells in a bioinformatic point of view, which express both markers of endothelial cells and one marker of cytotoxic cells, so either CD8 or gizmo. And this only occurred at day 5 in endothelial cells. So we first thought, of course, okay, this could be that some cytotoxic cells attach to endothelial cells, and then this would lead to cell death, but we have not found any markers of kind of endothelial cell death in these doublets. So what we rather think what this could reflect is very high amount of kind of endothelial cytotoxic doublets here at day 5 is the immigration of cytotoxic T cells and NK cells into the endothelium. So this is how the endothelium looks like in uninfected animals. So you have here endothelial cells. So this is actually platelets in the blood, some erythrocytes, and everything in dark violet are cell nuclei. And here at day 5, you have a much higher density of the cell nuclei. And the pathology says this is a clear indication of immigration of cytotoxic cells from the endothelium. So they come here, this is the bloodstream, and they immigrate through the endothelium into the tissue, and they start to eliminate infected cells. And so we have a very strong T cell and NK cell response at day 5. For example, this is seen here by interferon gamma expression. And we think that this doublet is a reflection of transmigration of lymphocytic cells through the endothelium, where there is some temporary attachment to endothelial cells. We don't only have a strong cytotoxic response, but also antibodies. So this in SARS-CoV-2 IgM ELISA. So already at day 5, we have very high levels of IgM antibodies, and this can also be seen in the proteomics, where we have an increase by about 2-fold. So this is log2 scale, 2-fold increase of IgM class antibodies. So what we observe here, and this has been also proposed for humans, that in a moderate disease course, you have a very strong Th1-driven immune response, which leads to rapid elimination of infected cells. So there's a very strong pro-inflammatory response triggered by the macrophages, what I've shown before, which then attracts all these T cells, and because it's kind of a healthy T cell response, it's self-containing. So, and the virus disappears, the inflammation disappears, and the animals get healthy again. Okay, so far I have shown this moderate self-limiting model of disease. But what about severe and critical COVID-19? I mentioned that we have this Roborowski Dwarf Hamster. There were actually 3 different kinds of dwarf hamster, Campelli, Roborowski, and Sungoros. And these 2 dwarf hamsters here, the Campelli and the Sungoros, they don't die. They also have a very moderate course of disease. And you can, for example, see this here. So this is body weight in the Roborowski Dwarf Hamster. The animal lose weight very quickly. This is red is standard dose, and this is low dose, and below 15 to 20% of body weight, the animals have to be killed due to animal care law. And this is not the case for the Campbellian Zungo's dwarf hamster. So we have this very, very unique exception here that the Roborowski dwarf hamster experiences a very fast and lethal course of disease. And of course, we are very interested to see now what is this one doing. So it's not ACE2 and it's not viral load. This would be the first thought. So this is an alignment of the ACE2, so this viral receptor of humans and different hamster species, and it basically looks all the same. So it's not that the ACE2 is kind of different and there is much faster uptake of the virus or so. And this is actually an asterisk is here. There's the differences in mouse. So what this essentially shows is that the hamster ACE2 is much closer to the human ACE2, and this is also the reason why you can infect hamsters but not mice with the standard human virus. Good, and it's also, so this is the viral load, for example, here, and it's essentially the same in the different dwarf hamster species. So this is not the reason why these Roborowski dwarf hamsters die so quickly. There was actually no genome available for the Roborowski dwarf hamster, and we wanted to do single cell sequencing, so we also had to make the genome. This was a very excellent collaboration with different people with top expertise in here. We did nanopore long read sequencing, Illumina short read sequencing, and RNA-seq. Worked very well. 60 contacts have more than 10 million bases, of which we have really a high quality genome. We can recover about 90% of genes. So I think now we are really at the moment, if you have a model, a kind of an animal model, but no genome, just make the genome yourself. For the Syrian hamster, there is a draft genome assembly available. We also are currently improving this. But now to the data, now that the last two slides of the talk. So we are especially looking at the endothelium. So this is to display an endothelial cell subtypes the way we like it in the single cell field. So there is bronchial endothelial cells. So they bring oxygen to the lung from the systemic circulation. Then there is the pulmonary veins and arteries. So this is where the oxygen flows from the lung, where it's being taken up from the gas exchange, which is happening in the capillary cells to the heart. So this is how it looks like. So if you have here these alveoli, for example, you have some capillaries here and here. So this is where the gas exchange happens through the AT1 cells, and then the blood goes to the pulmonary veins and then to the heart. The endothelium is very important in COVID-19. So this is a recent perspective, which thinks that there are some people who say that endothelial dysfunction, the immune thrombosis, is a key pathogenic mechanism in COVID-19. What does immunothrombosis mean? So triggered by reactions of the immune system, there is like microthrombi, and this can be very heavily observed in the Roborowski dwarf hamster. For example, you have these neutrophils releasing their neutrophil extracellular traps, so DNA, which then coagulates. There is activation of contact-dependent coagulation pathway, and in the end, all these blood vessels clog, and this also leads, of course, to severe lung dysfunction. This could be an important part of severe and critical COVID-19. So we focused also in our very initial comparison between the Roborowski dwarf hamster, so the one that dies quickly, and the Syrian hamster on the endothelial cells, and we have seen some very strong differences. For example, there is ICAM1, which is a cell adhesion molecule, and which helps transmigration of lymphocytes, which is only triggered by the standard dose, but not the low dose. But then, for example, you have differences. So CXCL10 is induced to a certain degree in Syrian hamsters, but not in Roborowski dwarf hamster. But on the other side, you have a very strong induction of CXCL11 in the dwarf hamster, but not in the Syrian hamster. These two genes are actually not only next to each other in the alphabet, but also genome. It's kind of like a gene duplication, but they can be clearly distinguished. And there is also the thing that the endothelial cells in the Roborowski dwarf hamster have a very strong expression of TNF-SF10. This is also known as TRAIL. and is one of the clinical markers of severe COVID-19. And so, of course, now we are interested, why do these cells, for example, have this high expression of this TNF-SF10 and these not? So this is now things that we can look at. Also interesting, CLCL7 and 8, which are lymphocyte attractants and which are somehow not triggered in the Roborowski Dwarf Hamster. One of the ideas that we are pursuing is that several of these genes are induced by interferon gamma, so there could be perhaps a very strong interferon gamma response in the Roborowski Dwarf Hamster, but not the Syrian hamster. Good, I summarize here. We think that Syrian hamsters can provide a full characterization of a moderate COVID-19 course. You have the strong transcriptional response by macrophages supporting this hypothesis that they are the key player in COVID-19. With this TLR NF-kappa B activation, only about, so the epithelial cells in the lung, they likely don't play a strong role in the entire disease because only few of them are actually infected and they don't react strongly. There is a strong cytotoxic and Th1 response in the Syrian hamster, and this is likely the reason why the virus is cleared out so quickly despite this transient inflammation. And of course, the big thing will now be the comparison of the Syrian and Photopus hamsters in order to nail down a bit the reasons for severe and critical COVID-19. So I have mentioned the people all in the beginning. I would like to thank them again, especially Geraldine Nouay and Jakob Trimper, Martin Witzenrath, and Andre Ott, who were like the key players in doing this. But as often in the case in COVID-19, these fast projects become only possible with like large teams of extremely dedicated people. And for that, I'm extremely grateful. Good, I'm done here. I would stop share and hand over to Herbert again.
Thank you very much, Emanuel. That was really a nice talk and very important data. So while the people think about their questions, I mean, to me, one really interesting thing is this behavior of the macrophages. So you mentioned that you think that's mostly a passive thing, while you are for sure aware of this recent Nature paper by the Chicago group, where they talk about this kind of that circuit between the virus-infected macrophages and the T cells, and they actually looked for a positive and negative strand there, and they conclude that actually in actually many of the infected macrophages the virus is replicating. So their model rather favors you know, the active infection of the macrophages. And then I was wondering, in your time course, you should actually, if the hypothesis is it's opsonization with antibodies, which you show is coming up at day 5 rather. So I was wondering, did you look into the earlier time points, whether the macrophages are already infected at day 2, let's say?
So yeah, yeah, it's a good point, and yeah, we know that the Chicago paper is really good. I mean, the RNA scope that they did is really amazing. So the question is a bit in which percentage of macrophages it actually replicates, and I think this is in the Chicago paper. The amount of negative strand RNA in the end was then relatively moderate. So most macrophages didn't have this. You know, I wouldn't exclude that. I mean, of course, I would never say there is no macrophage with replicating virus, but the general idea is more that it's not the big thing. Or perhaps, you know, like perhaps it replicates in 20% of the macrophages and so. But I would like to show this one here actually on that topic. And so here we have looked at gene expression in macrophages without virus here and with virus here. And as you can see, there is a strong increase. And this is also the case for ISG15. So if a macrophage takes up a virus, if a macrophage takes up a virus, so a lot of gene expression is being triggered. And this is actually in 82 cells, it's more the opposite. So for example, but I know that this data is not super strong, but for example, here at day 5, there is stronger ISG15 expression in 82 cells without virus. And this is actually what you would expect, because if you have a functional virus, a replicating virus in a cell, it has these very strong suppression mechanisms of interferon signaling. So if a cell gets interferon from outside or also the signaling pathways inside the cells are triggered and there's a virus in there, the signaling would actually be suppressed. And this is more the picture that you see here, that you have less expression of IST15 in cells with virus compared to those without. And so this is kind of the percentage having IST15, and these are in the cells having IST15, the expression levels. And you can see here it's a bit smaller here. And we think that this is more an indication that if there is productive replication in a cell, you have suppression of these pathways and lower expression. So, you know, in the end, I think one will have to settle for something. Yeah, it replicates in macrophages, but not in all of them.
Maybe not the prime mechanism for what you are saying anyway. So even if it's a mixture, then yeah. And then in this Chicago paper, they rather think it's a very low grade, like slow evolution of that. So macrophages migrate around and they are kind of spreading that inflammation by recruiting T cells. And they speculated, okay, so this is very early. And maybe the T cells that are recruited are cross-reactive. So they sort of maybe are not even trained in the course of that infection against SARS-CoV-2. And then since this is such a fast model you have, I'm also wondering the T cells that are actually being recruited by that CXCL10 burst by the macrophages. Do you think they are already trained? They are specific to SARS-CoV-2, or is it like any memory T cells coming in and boosting inflammation there?
So what you haven't done, and in hindsight, of course, we should have done this. We didn't do TCR profiling of the T cells with the lung, and I think this would be, yeah, I mean, this is essentially an experiment. I think that should be done, and which you're also proposing. We don't know, but given this rapid clearance of infected cells, I would say they have to be specific T cells, no?
Yeah, I'm also, I mean, I'm kind of wondering how that works, right? That it's working, that it.
Yeah, why is it working so well?
Right?
Yes, yeah.
So isn't that surprising? Because, you know, the adaptive response, at least from the textbook, takes a bit longer, I guess. Well, yeah, maybe I'm not correct.
You mean the T cell response?
Yeah.
Yeah, this is it goes very fast in the Syrian hamster. Yeah, that's true. But this is perhaps not the case in humans. Yeah, yeah. This is, you know, this is a topic that we are discussing heavily back and forth, and you know, like the matching of the disease course with humans. And the issue is that we don't have a lot of human data from very early time point in infection, which we have for the hamster. So this makes it a bit different. But of course, I mean, the hamsters is fine after 7 days, which is not the case for humans. I mean, even if it's a moderate disease, after 7 days, you still feel bad. Okay, perhaps I can go to the chat questions.
Yes.
Do the Syrian hamsters suffer with gastrointestinal problems during the first days of the infection? Why do they lose weight? This was by Heiner Schrieve. So as far as I understand, the veterinary people, it's they just, you know, like they eat less, essentially. They don't have gastrointestinal problems, you know, like they don't suffer from diarrhea or so. But we have not looked at the gut of these hamsters. I think there are people in New York doing this, but this might be something that might be worth, of course, looking as well, to kind of to also investigate the gut epithelium. Perhaps it also contains some virus. Yeah, so I cannot really only partially answer that question, unfortunately.
I think on the next one we touched a little bit, like how is that virus positivity correlating with the known entry factor?
Yeah, it's not correlating. It's more like anticoagulation. I haven't shown this here, but actually there's more virus in macrophages than in AT2 cells. But AT2 cells are more or less the only cells expressing ACE2 in the lung. So, and also NRP1, for example, is not. We don't see NRP1 as being somehow relevant. So virus is to a large part taken up by macrophages and to a smaller part by AT2 cells.
Yeah, and then Jörn Walter is interested in these doublets that you mentioned, or presumable doublets. So maybe you can elaborate a little bit on this.
Yeah, So this is this one here. So kind of so yeah. So when we looked at the data, we have started to see that what we identify as endothelial cells based on a set of marker genes at day 5, almost 60%, so this is fraction, almost 60% of these cells also contain kind of, you know, in the data table in Seurat, also have CD8 or Gizmo, which are markers of cytotoxic cells. Of course, it could be an artifact of whatever kind, but we have seen this only for endothelial cells at day 5, a bit for 81 cells as well, but for example, not from 82 cells. We of course first thought, okay, it's like something that happened during cell dissociation, but it's also more or less reproducible. So this is standard deviation from the 3 animals that we looked at. So I think it's kind of in a way real, but we don't fully understand it, I have to admit, because we don't really have antibodies against these hamster proteins. So what we would need to do is looking at these sections and staining for endothelial and lymphatic cells and really look close at like 100-fold resolution, whether there is really a strong interaction going on there. This is just derived from like general textbook knowledge. There is transmigration of lymphocytes through the endothelium into the tissue, and this is what we think this reflects.
That's a bit like the PICSEC, I think it's called, right? The paper from Omics lab.
Exactly. It was also a bit inspired by that.
Yeah. So Heiner Schrebe wants to know concerning SARS-CoV-2 infection during human pregnancy. Do you know of studies related to hamster embryogenesis and SARS-CoV-2 infection?
No, I don't know about this. No, unfortunately, I don't know about this, but of course it's something that might be of interest, yes. I know that there are several groups looking at human placentas. So there are, for example, placenta biopsies from pregnant women who are suffering or just had a SARS-CoV-2 infection. This I know, but I don't know of any animal studies of kind of pregnant animals.
Okay. Speaking of the hamster models, it's quite fascinating your plan to compare these 2 different closely related families, right? The Roborowski and the Syrian hamster. And so you mentioned this, the Roborowski dies very quickly, right? Mm-hmm. Then I was thinking, okay, but that's not exactly what we see in COVID-19 in the severe patients, right? It's more like a long-term thing. And along these lines, do you think if you age the Syrian hamster, you would maybe even get closer to human COVID-19 than with this Roborowski?
Yes, this is a good point. So so here on this slide, there was this initial publication here last year, age-dependent progression of SARS-CoV-2 infection in Syrian hamsters. And then they have indeed seen that the older hamsters suffer from much more severe disease. And of course, our first thought was, let's compare young and older hamsters, but due to, you know, like restriction on animal experiments, we couldn't do this because, yeah, you know, in our experiments, we kind of use hamsters that come out from like some drug or vaccination studies. So we thought of doing this, but unfortunately, we didn't make any progress there. But it would, of course, be something very interesting to do.
And I don't know, in the chat, we don't have any further questions. I noticed that the airway cells are really underrepresented in the single cell data looks beautiful. I have looked at many lung single cell data sets, so I think it's very, very nice. But it's apparent that you have a nice representation of 81, 82. So the alveolar epithelium, but the airway epithelium is quite a small population. Can you speculate on why that is?
Yeah, so I think generally we try to, I think it goes back into the standard problem of how do you dissociate tissue, and the lung is a relatively solid tissue. So, and then, you know, you don't want to digest for too long. So we did this 15 minutes at 37 degrees where we thought, okay, we get a pretty good single cell resolution, but of course you still deplete cells which are in very solid parts of the tissue. And this is why we think we get relatively few, for example, of these ciliated cells. So the 81 and 82 are the alveoli and also make up the most of the epithelial cells in the lung because there's this huge alveolar surface. I think it's like a football field for a human or so. But still, we also think that we deplete the ciliated cells in the course of cell dissociation. And this means that, of course, it would be best to kind of in parallel do single cell and single nucleus sequencing of the same tissue.
So you showed the macrophages are pro-inflammatory, the 82s not so much. Could it be that the airway epithelial cells in the early phase of infection, you showed a staining that it's like highly infected, that they actually also contribute, or?
Yes, there is on that. We haven't looked at airway endothelial cells, but there is a publication by people from Charité, from the Roland Iles group, who have extensively looked at, they did a lot of single cell sequencing on upper airways, airway epithelial cells, and we have reanalyzed that data as well. I don't have it in the presentation, unfortunately. But what we have basically seen there, you also have this strong activation of macrophages, which are likely immigrating macrophages in the upper airways, and also relatively limited activation of the epithelial cells. But still, you know, one aspect of moderate disease is that you have strong expression of interferon beta and lambda. And this has to happen at some point, and likely the upper airway epithelial cells are doing this kind of antiviral interferon signaling. But we also have the impression from reanalyzing that data that the pro-inflammatory signals from the upper airways come from the macrophages.
Interesting. All right, that was really great, Emanuel. Thank you so much. I think we are a little bit over time, so that in this respect, I would like to thank everybody for participating and Emanuel for a great talk. And next seminar in 2 weeks.
Thank you very much. Goodbye.
Thank you. Bye-bye, everybody.